right, now we're going to talk about tRNA. When we talk about tRNA, tRNA stands for transfer RNA. tRNA molecules are going to carry amino acids to the ribosome for incorporation into a protein. Now, when I draw my tRNA molecules, I draw them upside down, like the letter T. This represents my tRNA. Coming off my tRNA are nitrogen bases. So, these are going to be something like U, A, C. These are going to be carrying amino acids They're going to have specific shapes like this. They're transferring the amino acids. Now, when we look at the tRNA, it also has a 3 codon sequence. Now, we don't call these a codon. These are going to be called anticodons. So this is an anticodon. UAC that you see is one of my anticodons. So it contains 3 nucleotides that are complementary to mRNA codons. So these are always going to be complementary to our mRNA. So technically, you can be given your DNA sequence, TAC, CCC, GGG, to be easy. You then can figure out your mRNA sequence. AUG, GGG, CCC. Now, since the job of your tRNA is to transfer amino acids to the ribosome, technically, your tRNA that would be complementary to the mRNA, these are anticodons, would be UAC, CCC, GGG. So these are anticodons that are transferring the amino acids. If we were using our genetic code chart, we then can figure out which amino acid using our mRNA is actually being transferred, which we know if this is an anticodon to AUG, then the amino acid it's going to build the protein would be methionine, because we're using this to figure out your amino acid. CCC encodes for amino acid pro. I already made a mistake. This should say GGG. GGG encodes for the amino acid glycine. And these would have peptide bonds in between. So now we're adding that extra step with tRNA, which has anticodons, with mRNA. But we need to see how that works in the terms of a ribosome. So when we look at a ribosome, the ribosome is the location where translation occurs. This is where translation occurs. When we say translation, you're taking the mRNA and it's being read and it's being transferred into a protein. So the job of the ribosome is to read mRNA, and the second thing that the ribosome does is it has to form peptide bonds between amino acids to build proteins. Now, it does have an enzyme. The enzyme peptidyl transferase is the enzyme that is going to build your peptide bonds. Now, another thing that we talk about when we look at the ribosome is that it has 3 major areas. But before we do that, the first thing that we need to talk about is mRNA. So mRNA is going to attach to the ribosome, and the ribosome has 3 distinct areas. Or multiple tRNA binding sites. All right, the site right here is called the E site. The site in the center is called your P site. And the site over here is called your A site. So let's write some notes on here. So this is the ribosome. The ribosome has multiple tRNA binding sites. So for each one of these sites, you're going to have a tRNA that is going to bind. So if we follow the letters that we had on the last one, AUG, GGG, CCC, you could write those in. AUG, GGG, CCC. Those are your codons. So each one of these binding sites are basically for your tRNA. So the anticodon for this one would be UAC. This one would be CCC, the anticodon. And this one would be GGG, anticodons. So anticodons are complementary to your codons. So this one would literally be GGG. The anticodon over here would be CCC. Ran out of room a little bit. So we have 3 binding sites for tRNA. Now I'm not done with this just yet. All right, the first one I want to start with is your P site. P site. This is going to bind tRNA attached to the growing protein or polypeptide. Your A site is going to bind the next tRNA, which carries, again, the next amino acid. And your E site is going to bind tRNA that carried the last amino acid. So the way I remember this is it always is moving from left to right. I want you to think of E, the E site, is where it's going to exit. So it no longer has its amino acid anymore. Now we know that AUG is the first amino acid. So over here, the growing peptide chain, methionine was the first one. Well, CCC is proline. So this one is literally going to have proline attached to it, and bound to proline is methionine. So the P site is growing your protein. The P site's growing the protein. And then the A site is going to bind the next amino acid. GGG encodes for glycine. So this is the next one that's going to be incorporated and bound to proline. So AUG was the first amino acid because we know that this is the start codon. This was the start codon, so it was the very first one read. So the first amino acid was methionine. So AUG moves to the E site and it's going to exit. The tRNA no longer has methionine attached to it because it already transferred it. So your P site is reading GGG, so the anticodon CCC is going to transfer proline to methionine. It's bringing it to the amino acid. And then this one is the third one. So this is going to be the next amino acid that is going to get added to the growing peptide. So your ribosome has 3 sites that are binding for tRNA, which are transferring it. Your E site is going to exit, your A site is for the next amino acid, and your P site is for the growing protein. Now we can use translation and discuss it in the same terms as transcription and translation. We can say that there are translation steps. The first thing is initiation. Initiation for translation is going to require mRNA with a start codon is going to initiate translation. So, your start codon is AUG. AUG binds to the ribosome and we start translation. The next thing, elongation. To elongate the protein, you're going to have to involve amino acids. So the way we do that, tRNA binds to the A site. If its anticodon is complementary to the mRNA codon at the A site. Peptidyltransferase is going to form a peptide bond in order to elongate the growing amino acid chain or protein. And the third thing is termination. It has to stop somewhere. So how we terminate it is going to have to be a stop codon. So elongation continues until the ribosome encounters a stop codon. And we have 3 stop codons: UAA, UGA, and UAG. When it encounters a stop codon, we terminate it and we release the protein, also called the polypeptide, from the ribosome. It all goes back to your codons and the genetic code. When we hit a stop codon, UAA, UGA, or UAG, we stop translation and we release the protein from the ribosome, and translation is over and we've created a protein.